Hey everybody, and welcome back to Bio229 Learning Remotely in 2020. We have been working on Chapter 3, which is basically visualizing bacterial cells and then talking about the parts of the cell that we might actually visualize. I am outside right now recording this lecture because inside my house it is a little on the noisy side right now. So in advance, apologies for chipmunks and squirrels and a variety of noises that you might hear, maybe a little traffic too. And if my neighbor who is building a stone wall and cutting stones starts the saw, you'll be the first to hear about it. So anywho, let's get back to microbiology. I'm your host, Mary Annarella, and here we go. We left off getting into the parts of bacterial cells. We'd already talked about groupings, shapes, so strepto versus staph flow, and bacilli rods versus spheres, and spirochetes. And we left off last time talking about the cytoplasmic membrane. Now we're starting to get into the actual structures of bacterial cells and differences between the structures of eukaryotic cells versus prokaryotes. So both prokaryotic cells and eukaryotic cells obviously have a cytoplasmic membrane. Okay, so they're made of phospholipids. We've talked about this before. This is all review for you all. So we've got a phospholipid bilayer here, proteins that are embedded. That's what these little brown lumpy thing, this brown lumpy thing and this blue lumpy thing are. And it's a fluid mosaic model of the cytoplasmic membrane, meaning that it's very flexible. It's not rigid at all. So the functions of the cytoplasmic membrane, well, it's to keep the outsides out and the insides in. I mean, if something's going to be a cell, it needs to be, we have to have a defining spot for what's inside a cell versus outside the cell. But this second bullet's really important here. So let me take a screenshot for you all. We'll do yellow. Okay. It's very important in transforming energy into usable form for the cell. So the electron transport system, if a bacterial cell has an electron transport system, it will be embedded in its cytoplasmic membrane. So this is for making ATP. Right, so if bacterial cells, if there are prokaryotic cells that do electron transport, that electron transport system will be embedded in their cytoplasmic membrane. Not the case for eukaryotic cells, and we will get to that again too. All right, and it's important for transporting molecules in and out of the cell. All right, so we got to talk quickly about transport across the membrane. A lot of substances will just simply diffuse across the membrane. This is passive. It does not require energy. And again, this is all review for you. So passive transport does not require energy. Active transport does require energy, right? So active transport does require energy to make it happen. And that happens too. We'll talk about that in a minute. Okay, but diffusion is an example of passive movement. So it's movement of a solute from an area of high concentration to an area of low concentration. So it's like, if you have a glass of water and if you drop a little bit of food coloring into it, let's say yellow food coloring and blue water, here we go. You know that it's not going to look like that if you sit around for a while. And you also know that this food coloring is made up of, I'm putting little dots here. It's made up of a bunch of molecules that are tightly packed together. You give them room to spread out, they're going to move from an area here where they're highly concentrated, moving out here to where they are less concentrated. So by the time, this sits around, and people are like, well, they're going to spread out. They're going to diffuse. Yeah, but what does that mean that they diffuse? It means that they move from an area of high concentration here out to here. They take so that all the molecules can take up as much room as they possibly can. This is passively, it happens all by itself, and it does not require the input of energy. And that's how things will move in and out of a bacterial cell, too. If there's a whole bunch of stuff outside the cell, and if it's the kind of molecule that's allowed to simply diffuse across the membrane, something like, I don't know, oxygen, a bunch of them will just move right in until that gradient has been equalized. And this is just, I have here for small molecules like water, oxygen, CO2 will just diffuse across the cell membrane passively. Passively. But not everything can just, take another screenshot of this one. Here we go. Let's get a different color. All right, things like oxygen or water can just push these phospholipid molecules out of the way and diffuse right in. No big whoop. Some molecules need a little help doing that, even though it's still technically diffusion. A protein will help facilitate the diffusion. So this is facilitative or facilitated diffusion. It's just when a molecule, like here you've got a whole bunch of these green molecules outside the cell, and they're allowed to diffuse through, but they can't just push these phospholipid molecules out of the way. A transport protein will help them out with that until there's an equal concentration both outside the cell and inside the cell. We say things have been equalized there. So it's just facilitated diffusion. All right. But not everything can diffuse across the membrane. All right, we say that membranes are, I'm trying to write on this. I got to take a screenshot first. And by the way, have you all noticed that I actually started this off in landscape? I'm so darn proud of myself. You be too. It's the little things, people. All right. Not everything can cross the membrane, right? We say that the membrane is, in this case, yellow's not working. We say that it's a selectively permeable membrane. That means not everything can cross the membrane by diffusion. Some things can cross passively and some stuff can't. And for the things that can't, you need active transport, and that requires energy. So this is active movement, requires energy to move substances against a concentration gradient. And it takes energy in the form of ATP, or what we call a proton motive force, or a PMF, which you know what that is, you just probably don't know that it's called that. And this is just for expelling waste, secreting proteins, blah, 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 blah. Okay, let's explain this stuff. What is this? Active movement requires energy to move substances against a concentration gradient. I don't know what that means. Let's talk about what that means. For that, we'll go back to notability. All right, we're doing purple here. All right, when I did the little glass of water with a blob of, well, now we're in purple mode. Purple food coloring here. You have a high concentration here in the little blob of food coloring and a low concentration out here. So there's a low concentration of dye out here in the water. There's a high concentration in the blob. This is what we call the situation where there is a high concentration of a substance in one area and a low concentration of it right next to it is called a concentration gradient. It's having a difference in concentration of a substance from one area to the next. And here is the deal. Concentration gradients are not stable in nature. It is just one of the laws of physics. It's just the way it is. It is an observation that these are not stable. And sometimes they say things like, nature doesn't like a concentration gradient, and I'm just abbreviating conc grad there. Nature does not like a concentration gradient because it's not stable. Nature wants it to be equalized. Wants it to be equalized. That's right, you just saw me spell a word with an equal sign. All right, so you know if we let this sit around here for a while, this glass of water, all those purple dye molecules are going to spread out, and there's not going to be a high area of concentration and a low area. Everything will be equal concentration. So there is no gradient here. We say the gradient has been equalized. Right? We say this is like when these molecules diffuse to equalize the gradient, we say it's going down the gradient. Down or with a gradient. Kind of like you're going downhill. Right? Well, let's get a different color for the hill. Okay, a hill is a gradient, right? Like if you're at the top of Mountain Road in East Hampton, up like near where the log cabin is, and you go down that road, there's a sign that shows, you know, steep hill ahead, or sometimes it will say steep grade. Steep grade. A hill is a gradient. We have an area of high elevation down here and low elevation down there. And you know darn well that if you were riding your bike down here, down that hill, I can't draw a little bike. Let's put a person on there. You know you are not going to have to pedal to go down. It takes zero energy to go down that hill. It takes no energy to go down a gradient, right? But if you were going to go back up a gradient, go against the gradient, that's going to take you a whole lot of energy, right? So think up here to our purple dye. Well, now I should go back to purple, huh? To our dye molecules up here. Have you ever seen a bunch of dye molecules that have, you know, diffused out, and, you know, you have this pale purple water? Have you ever seen it, them all get back together again? Getting the band back together, getting the dye molecules back together like that? That does not happen by itself, right? This would be going down the gradient. You can do this. You can do this, but it takes energy. This would be active transport. That color does not show up very well, does it? Active transport. It's like going uphill. Of course you can go uphill, but it takes energy to do that. So active transport involves moving a molecule against a gradient. And it's for stuff like, you know, what if all these waste products accumulate in a cell? They got to get out, right? And even if they can diffuse, you'd be like, okay, we've got 6 inside the cell and a concentration of 0 outside the cell, so 3 of them are going to move there. So we'll move them out. There we go. Now the gradient has been equalized, and these ones aren't going to be able to go because that would make it worse. But these are waste products, so you got to get them all out of a cell. So in this case, it would be, these last 3 would be moving against a gradient, and that would take energy. Took energy to do that. Okay, so yeah, it's for expelling waste, secreting proteins, things like that. Because sometimes you just got to do active transport, and sometimes you just got to spend a little energy. You're hearing all these birds back here? I do not know what kind that is, but it is making a racket. All right, I do want to mention that active transport is done via a protein. Proteins help them cross through. And I did want to mention group translocation, and it's when a molecule is chemically changed, like, some other molecule will be added to that molecule that's being moved across the membrane to change what it is, and so it kind of tricks nature that way and defies the gradient, but it still takes energy. All right, so now we're getting to some other structures. So now that we've done the cell membrane, let's talk about a few structures that are on the outside of prokaryotic cells. All right, capsule and slime layers. Wow, doesn't that sound exciting and fun? A capsule or a slime layer, I'm going to be able to write this down here. It's generally made of polysaccharides. It's a polysaccharide layer that surrounds a bacterial cell. So like right here, and the cell membrane and cell wall will still exist, but it'll have this diffuse polysaccharide layer around it. And the only difference between the capsule and a slime layer, they're made of the same stuff, is that the capsule, I have up here on the PowerPoint, you can read it, it's distinct, the slime is diffuse. The polysaccharide molecules in a slime layer, are farther apart. So it's just looser. So kind of like Jello versus agar, that would be harder and distinct. They use it for attachment to other surfaces, meaning maybe an inanimate surface, or even your cells. So I have here Streptococcus pneumoniae. Only the form that has a capsule is actually pathogenic, is infectious. It's much more infectious because it can stick to the cells lining your lung much more readily that way. Slime layers, they help bacterial cells, attach to a hard surface. And also, I should add here biofilms. Biofilms are the same stuff, but it's not just surrounding one bacterial cell, it's surrounding a whole bunch of them. And wow, I put this iPad on, did I? Yes, I did. This is on airplane mode. Why did I get a notification there? Anywho, sorry about that. That was an Instagram notification. All right, biofilms will generally attach a whole population of bacteria to a surface. So every now and then, when there will be like a lettuce recall, it's usually because there is some sort of pathogenic bacterium, often E. coli, that will have formed a biofilm, and you can't wash that off. It's not going to wash off with water. So they're hard to get rid of because they really help a population of bacteria attach to food in that example. All right, flagella are on the outside of bacterial prokaryotic cells as well. They are used for locomotion, or sometimes we say taxis, movement. They'll whip around and they help bacterial cells move. They use a proton motive force to power it. And I want to talk about what a proton motive force is. That's what the PMF stands for. So let's go back here. Proton motive force. Motive force. Hugely important in how cells make energy and use them, but proton. Okay, a proton is just a hydrogen ion. If you recall from Bio229, the hydrogen atom, and I know they don't have a membrane to them, but I got to draw a circle or something to show you that there's a hydrogen atom here. In its nucleus has just one proton. I'll put a P+ there for a proton, and no neutrons. So it has a proton in its nucleus and one electron outside of its nucleus, buzzing around there. You learned all this in a previous class. But this electron here is kind of loosey-goosey, and it can exit the building. And then all that you will be left with is one proton in the nucleus and nothing else out there, because most hydrogen atoms don't have any neutrons to them. But this becomes a hydrogen +1 ion, right? But we can use the term proton for this one, and it's the only atom we can do this with because it's only got one proton left in its nucleus. There's nothing else there when it loses its little electron. So this is a proton. A proton. So now we know what a proton is in a proton motive force, or PMF. If you recall from learning all about how cells make energy through cellular respiration, you got to this thing in the electron transport system where, okay, and in that case you're in the mitochondria. We wouldn't be there in a bacterial cell. Let me draw a bacterial cell. You've got these proteins. Oh dear, how did my battery get so low suddenly? I looked at it a little bit ago and it was at 40%. Ack. I guess I need a higher battery for that. Okay, before I gotta stop this and run off to get my charger, we'll talk about protons that come from the sugars that we feed bacterial cells that get pumped across the membrane. They get pumped across the membrane, and that sets up a concentration gradient. This is a concentration gradient. When a whole bunch of these protons get pumped across the membrane, and in eukaryotes it's the inner membrane of the mitochondria, they osmose back through ATP synthase, right? They osmose back through, and that causes ATP synthase to basically turn and gives ATP synthase the energy to put the phosphate group back onto ADP to form ATP. Only in the case of a bacterial flagella, instead of ATP synthase, it'll actually use a flagella, and it'll make the flagella whip around. So let's envision this as a bacterial flagella. This is called a proton motive force. It's the flow of protons across a membrane to generate energy. All right, I'm going to go get my charger and come right back. Oops. Okie doke, and we're back. All right, so flagella, structure on the outside of a prokaryotic cell. They're used for movement, powered by a proton motive force. Bacterial cells could have one, they could have two, they could have three, they could have a ton of them. So let's take a look at a few. Here I've got one with just one. You don't need to memorize monotrichus or amphitrichus. No worries there. This one in the lower right-hand right-hand corner is actually E. coli. E. coli's got a ton of flagella on them. We're not going to see any when we stain bacterial cells this week. Okay. So here we go to the next one. All right. Other things are pili. Pilis are just basically smaller version of flagella. So if I take a screenshot of this one, okay, you can see some pili here, but like if this is a flagella, pili are going to be shorter and thinner. They also help cells to adhere to other surfaces, but not in a way like a capsule or a slime layer does. That's just from stickiness. Think of these as little hairs almost. And they are made of protein, so that's not too far off from hairs. We've got movement on a solid surface. Pili can actually help them move. And there is one special pilus. Pilus is the singular version of pili, called the F pilus, which is involved in bacterial conjugation. Bacterial conjugation is the exchange of DNA between two bacterial cells through this pilus. Exchange of DNA between bacterial cells through that pilus. And here we've got a picture. Okay, yeah, here we go. This is the F pilus here. It looks about as big as the flagella. The F pilus is an exception. These other things are just little tiny pili. Right there. And we will learn more about the F pilus, which is also short for the fertility pilus, later on in the course when we get dig a little deeper into conjugation. Alrighty, other things that are inside the prokaryotic cell. Well, you got the DNA, which is just a circular chromosome. Some storage granules. Got a picture of an endospore for you, where the cell basically just, kind of goes into hibernation. Having some water here. All right, but the ribosome, so there are a couple of differences between the DNA and the ribosomes of a prokaryotic cell. So just to go ahead and show you the DNA, it's a circular chromosome. No, you know what? I'm not going to do it this way. I'm going to go back here. So prokaryotic DNA. It comes in two forms. You have the chromosome, the bacterial chromosome, and it's not in a nucleus, and it's in a nucleoid region. It's circular. So if this is our bacterial cell, we've got a circular chromosome like that. But there is also other DNA in bacterial and in prokaryotic cells called plasmid DNA. Plasmids are small circular pieces of DNA. Like that. They contain genes too. Chromosomes contain genes and plasmids do. And not all bacterial cells will have the same plasmid. There are thousands of different plasmids that have been isolated, right? But basically everything that a bacterial cell needs to survive and every protein that a bacterial cell needs to survive has information in its chromosomal genes. So we call these housekeeping genes. It's everything that's necessary for survival. Whereas genes that are on plasmids, are usually for enhanced traits. Traits that aren't necessarily required for survival. And an example of an enhanced trait, a gene that's on a plasmid, is antibiotic resistance. So genes that give a bacterial cell resistance to a particular antibiotic. So for example, there's an antibiotic that we use in the laboratory. We actually don't use it for people, but it's called ampicillin. And there is an AMP resistance gene. So the superscript R up here is for resistance. And it's on a plasmid. If I draw a plasmid a little bit bigger, here we go. If I draw the gene, the location of a gene is a little box on this plasmid. and this is an ampicillin resistance gene. Okay, I'll draw my little plasmid up here. This bacterial cell, if it's got this ampicillin resistance gene on this plasmid inside of it, it will be resistant to the effects of ampicillin, which means I want to draw a fist and write resist. It will resist ampicillin. It won't be killed if it's exposed to ampicillin. So if something is resistant to an antibiotic, that antibiotic won't kill that cell. And I'm just putting it out there because resistant, there we go. Sometimes students have gotten hung up on the language before. Like people are like, oh, well, if the bacterial cell is ampicillin resistant, that means ampicillin will kill it. Nope, opposite. We say if something is sensitive to an antibiotic, that antibiotic will kill that cell. Antibiotic will kill that cell. And my neighbor has just started mowing their lawn, so we're going to move this party indoors, where I think it's probably a little quieter now. So come with me on my journey and I will say more about resistance and sensitivities. It's kind of like if somebody is sensitive to gluten, it means that gluten makes them feel yucky, right? And if a bacterial cell is sensitive to an antibiotic, that antibiotic is going to make them feel crappy. It's going to kill them, in fact. And we like to use that. DNA that is inside prokaryotic cells can be chromosomal. Those are for the housekeeping genes. Or they are often loaded with plasmids. Alrighty, ribosomes. Ribosomes are different in prokaryotic cells compared to eukaryotic cells. Here we go. Okay, we already talked about prokaryotic DNA. Got it. Ribosomes. Ribosomes are made up of two parts. There is a large ribosomal subunit and a small ribosomal subunit. And in prokaryotes, the sizes of these subunits, and it's important, here we go, are, say, 30S and 50S. So over here, here's our 30S ribosomal subunit. This is the small one, and the 50S ribosomal subunit. When you put them together, here, you get a 70S ribosome. Yes, I know that 30 plus 50 does not equal 70. It equals 80. But that's not how this math works. These S's, this 30S and 50S, this S stands for Svedberg. And Svedberg, I don't know if I spelled Svedberg correctly there. Maybe it's a U instead of an E. Svedberg is a scientist who figured all this stuff out. And actually, I think he's the guy who invented the centrifuge. And so Svedberg unit is a measure of how a molecule will move through a sort of liquidy medium in a centrifuge. So it has to do with size, weight, as well as shape. And when you put the 30S and the 50S together, the shape that it gives them, it will move basically 70 units in a centrifuge, not 80. All right, so don't get confused about that. Maybe I'll put red. Do red right now. So bacterial ribosomes are 70S. Eukaryotic ones are 80S. they've actually got a 40S plus a 60S, which, yes, does not add up to 80, but that's how this math works. And I'm pointing this out because this is a big, important difference between eukaryotic cells and prokaryotic cells, because a lot of antibiotic drugs that we use will target the 70S ribosome. So if you take an antibiotic that will destroy cells that have 70S ribosomes in them, they'll leave your cells alone. So that's really, really cool. And actually, this is a good time to talk about this. Delete that screenshot. Okay, so ribosomes in prokaryotes are 70S, which is 30S plus a 50S in prokaryotes. And they're 80S in eukaryotes, which is a 60S plus a 40S. So these are eukaryotes. Prokaryotes over here. I'm going to erase that. So it's a big hairy deal there. Oops, that's not what I wanted to do. Okay, a lot of antibiotics. Some antibiotics target 70S ribosomes. And if you destroy 70S ribosomes, that prokaryotic cell can no longer make protein, right? Because you already know, again, from previous classes, that ribosomes are protein factories. They do protein synthesis. And if you destroy the ribosomes in a cell, that cell is not going to be able to make proteins, and that cell's not going to be able to function, because you need proteins to be able to function. Every cell needs proteins to be able to function. So antibiotics target 70S ribosomes, but a major class of antibiotics targets peptidoglycan. Either its synthesis or just will break up peptidoglycan itself. So that's a cool thing, because think of it this way. Well, maybe I don't want to make us green. Let's do blue people. We'll pretend like we're Avatar. Here is a person, and they are sad because they have a bacterial infection. I don't know, somewhere on their arm, whatever. Well, the bacteria have peptidoglycan and 70S ribosomes, whereas the human cells have 80S ribosomes and no peptidoglycan. So antibiotics have to take advantage of differences between your cells that we're trying to keep healthy and alive, and bacterial cells, prokaryotic cells, that we're trying to get rid of if they're causing you an infection. So if you give someone a drug, an antibiotic that destroys peptidoglycan, these guys are going to have a really hard time growing, right? So that clears the infection. Two major, I just removed his arm, or her arm, or their arm. I just removed their arm. Here, let me give them hands and feet. Make them feel better. I'll give them hair, too. I don't like that hair. I'm going to give them a bun. Bun right on top of their head. There we go. And they're not sad anymore because we just got rid of their bacterial infection, and now they are happy. There we go. So, classes of antibiotics, they tend to target, well, actually most of them, this is the biggest class, targeting peptidoglycan, but a lot of them do target 70S ribosomes. Okie doke. Moving on to eukaryotic cells, because eukaryotic cells have some things that bacterial cells don't. Starting with their plasma membrane, with their cytoplasmic membrane, it's so similar to prokaryotic ones. It's got a phospholipid bilayer, it's got proteins stuck in it, but the bilayer is asymmetrical. That's a big difference. And you know how I have you all starting, keeping a running list of the differences between prokaryotic cells and eukaryotic cells? This is one thing for it. Well, actually two things for it. And to show you what all this means, okay, what this means is that the bilayer is asymmetrical. It means that the outer layer and the inner layer, okay, you've got all these polar heads here and the hydrophobic tails that are the fatty acids that are hydrophobic. They are different from each other. So we say that it's symmetrical. They have different fatty acids between the inner layer and outer layer. That's not the case in prokaryotes. Prokaryotes have an asymmetrical plasma membrane. So it does not want me to do that. Get rid of that. Didn't want to come back up. There we go. Okay, so plasma membrane is asymmetrical in eukaryotes, and it is symmetrical. Asymmetrical does not look like I spelled it right. Asymmetrical? I don't know. Symmetric. Oh, there are two M's. Well, dang. Asymmetrical. That does not look right. Oh my gosh, it's a good thing I'm not teaching English, right? It's symmetrical in prokaryotes. And also, in prokaryotes, the cell membrane is used to make energy. Not the case in eukaryotes. In eukaryotes, the cell membrane is not used to make energy. So some big differences there. Right. Transport across the membrane, same deal. You get diffusion. There's facilitated diffusion in eukaryotes. They do active transport the same way. They'll have little protein channels that'll let small things in and out. But there are two major things going on in eukaryotic cells that do not happen in prokaryotic cells, and that is endocytosis and exocytosis. So endocytosis is bringing in material from the environment, and exocytosis is spitting something out of the cell. So endo bringing in, exo going out, exit, exocytosis. And let's just take a look at that. Okay, so for endocytosis, well, I'm just going to skip right ahead here. So this is endocytosis bringing stuff in, so we're talking endocytosis. Okay, there are two forms of it. One down here, phagocytosis, that does not show up. Phagocytosis, and two, pinocytosis. What color would work better here? Let's go with red. So with phagocytosis, you end up with pseudopods forming around solid, large material. So think of it as, as a large granular food substance, bringing it in, and it forms a lysosome that we call a phagosome around it. And yeah, it'll fuse with a lysosome in the cell, but it's basically a little vesicle, the phagosome. Whereas with pinocytosis, this is for bringing in substances that are in solution. So sometimes people are like, oh, phagocytosis, that's cellular eating. Pinocytosis is cellular drinking. Yeah, kind of, sort of. So molecules in solution. And it'll form what we call it an endosome. It's just a vesicle. Oh, I can't spell vesicle. Just call it an endosome. And here, exocytosis. With phagocytosis and pinocytosis, the cell uses its own membrane to surround the substance to bring it in. So this is made up of membrane here, these vesicles. Likewise, when they spit something out, they'll make a vesicle, in the Golgi apparatus, whatever. It will actually fuse with the cell membrane and then basically make a cut in the cell membrane here. So this is actually, this red part here that I'm drawing was actually this here. So the vesicle will actually fuse with the cell membrane and then release this substance to the outside of a cell. There we go. Okay, let me back up. Okay, so pinocytosis, something is encapsulated, brought in. Pinocytosis, phagocytosis, cells forming pseudopods, those false arms to grab a particle. Oops, no, what am I doing there? Exocytosis, this is for secretion of proteins or sometimes even waste products. All right, the eukaryotic cell has a cytoskeleton that gives structure to it. The prokaryotic cells don't have either. And it's actually the cytoskeleton that rearranges itself to support the cell's needs. And one of the times that it does rearrange itself is during mitosis. So let's take a picture of that. Whoa, where'd we go here? All right, so if we've got a eukaryotic cell here, and here's the nucleus, and here are some mitochondria, for example. And here you're going to have, these things aren't just floating all around, if you've got, the endomembrane system and whatnot. There are proteins that help attach them to each other and to the cell membrane. And you have 3 different types of proteins here. You have these microtubules, actin filaments, and then intermediate ones that will, kind of crisscross things like that. But it's like a mesh network of proteins all throughout the cell that give it its shape. And when a cell actually divides and forms the mitotic spindle, does that look familiar to you? And you've got all these chromosomes that are like pulled to the metaphase plate and all that. These things in yellow here, the mitotic spindle, those are actually microtubules from the cytoskeleton that have become the mitotic spindle. So I liken these to this toy called Kinects. If you want to Google Kinects, you can build the Eiffel Tower. You can build a dinosaur out of one, like a T-Rex, and then you put a motor on the thing, and you've got a T-Rex made out of all these sticks and the like. But a kid will build something out of it, and then you take it apart, and then you can build something else. And that's the exact same thing with the actual cytoskeleton, that it can disassemble itself and rebuild itself into a new shape. So it changes to meet the cell's needs. And it's made up of these 3 parts. Microtubules, you need actin filaments, those are the longer pieces, and then these intermediate filaments are the shorter ones. So there we go. We are, come here. Why is this not working? Well, this is unusual. Let me take a screenshot and see if it'll just enlarge. Yes, no, but it's blurry. Crap. I'll just edit this out later. Go to Dropbox, reopen Dropbox. Okay, here we go. Oops, nope, that didn't work. Okay, here we go. So if we take a look right here, all right, we've got a mitochondria here, we've got our microtubules, actin filaments, which are longer, and these intermediate ones, which are like these little connector ones. So think of those as like the shorter connects. You do not need to know like the structure of the intermediate filament, but you do need to know actin, intermediate, and microtubules. All right, eukaryotic cells have flagella as well, only they don't have pili. So there's a difference between a flagella that's in a bacterial cell and a eukaryotic flagella, and that is this. I mean, they have the same arrangement, and again, I don't really care that you know that the 9+2 tubule construction of it, but I want to point out that, see this covering here? This is the cell membrane. And it covers the entire flagella. So technically, a eukaryotic flagella is inside a eukaryotic cell, whereas a prokaryotic cell, the flagella is actually on the outside because it is not, the prokaryotic flagella is not covered by the cell membrane. So that's another difference to add to your running list. There. And it's the same deal. Well, okay, bacterial, let me do that. If you have a bacterial cell with flagella, that flagella is covered with cell membrane. So it's technically inside, and it's the same deal with cilia, which are just shorter versions of flagella, but they too are covered with cell membrane. So cilia are inside. By the way, so no pili in eukaryotic cells, and there are no cilia in prokaryotic cells. If I say, oh, there's this bacterial cell with cilia all over it, nope, doesn't have that. This looks like a brontosaurus or something. That is like the most ridiculous thing I think I've ever drawn in my life. It's a dinosaur. Alrighty, so inside the eukaryotic cell, there is a nucleus. You already know that stuff. We've got supercoiled DNA with histone proteins. I talked about histones last time. They are proteins that chromosomal DNA is wound around, and they play a role in stabilizing eukaryotic chromosomes, but they also play a role in gene regulation, turning a gene on and off. I want to point out that the nucleolus right here, let's get it a little bigger, is where ribosomal RNA is made. Ribosomal RNA, or rRNA, is made there. It's part of the ribosome. The ribosome is made up of RNA, actually, and it's made in the nucleolus. There are nuclear pores where things get in and out. Got that. Yeah, and it houses the chromosome. And I already showed you this last time. This is just an this is an electron micrograph of DNA, and those little grainy things are histone proteins that they're wound around. It's so cool. Love it. All right. Inside the eukaryotic cell, you will not find, inside the eukaryotic cell, you will find membrane-bound organelles. And one of those is the mitochondria. So you will not find them in prokaryotic cells. So the mitochondria. Actually, I should do it in orange. books always code the mitochondria in orange. Well, actually, all right, if this is our eukaryotic cell and you've got the nucleus here and you've got your nucleolus there, whatever, you've got these little mitochondria. So this is the mitochondria. It has two membranes. So if we blow this up, and I draw it like that, it's got an outer membrane and an inner membrane that's very highly folded. Again, this is all review for you all. So I'm trying to just buzz through this pretty quickly. And the folds are called the cristae. And the outer membrane is smooth. The cristae, these folds, give the inner membrane way more surface area. Okay, and in here, this is called the matrix of the mitochondria. It's just the space inside. And inside the matrix of the mitochondria, you have some stuff. You've got little pieces of DNA, and you've got ribosomes. So up here, I'll draw some ribosomes in there and DNA in there. Alrighty. Well, people were looking at this going, this is interesting. This mitochondria, it's about the same size as a bacterial cell. That's one thing. Well, someone's like, well, check it out. It's got DNA in there. Let's look in there. The DNA, they sequenced it, and it matches up to just some general prokaryotic DNA sequences. So DNA, or rather mitochondrial DNA, matches prokaryotic DNA. And then finally, someone's like, all right, we got to check this out. This is freaking weird. Let's look at the ribosomes. And they found out that the ribosomes are 70S ribosomes. So they're the same size as a bacterial cell. Their DNA matches prokaryotic DNA, and they have the same size ribosomes as prokaryotes. If you're thinking what I'm thinking, this is the evolutionary result of a long ago ingested bacterial cell. So it's an ancestor of an ingested bacterial cell, from way, way, way back. And this theory that the mitochondria came from a long ago ingested bacterial cell, some eukaryotic cell with a nucleus ingested a bacterial cell and made it its very own, is called endosymbiosis. Endosymbiosis. And the three reasons that we believe that the mitochondria is the result of a long ago ingested bacterial cell, because it's the same size as a bacterial cell, but that's not really much evidence there. But 2 and 3, these are the big deals here. Mitochondrial DNA matches that of prokaryotic DNA. Come on, Mel. Come on. Oh, my dog wanted up on the couch. I had to call her up there. She looks at me like, do I have permission? Can I climb up here? Like, yes, you're allowed on the couch. Come on. And they have 70S ribosomes too. All right. And this is all about making ATP. These are ATP factories. ATP factories. Alrighty. Another membrane-bound organelle that some eukaryotic cells have, not all, is the chloroplast. Chloroplast is similar to the mitochondria in that it has two membranes, but it's kind of a lumpy shape. So it still has two membranes, an outer and an inner, and the inner one is folded, but they're like stacked, like accordion folds. Somebody said once, oh, this looks like a resistor or something, like the inside of a radio. I'm like, that's exactly what I was going for there, because this thing really does function like that. The stacks aren't called cristae, though. These are called the thylakoid membranes. And this isn't the matrix, this is called the stroma. And what's interesting is that in the stroma, there is, guess what? There is DNA, chloroplast DNA, and ribosomes. So guess what? Well, actually, these ribosomes are 70S. This whole thing is about the same size as a bacterial cell, and when they sequenced chloroplast DNA, so let's say chloro-DNA, sequences not only match just general prokaryotic DNA, it's almost an exact match to a bacterial cell that is around today, and that is called cyanobacteria. So chloroplast DNA matches cyanobacteria, which used to be known as blue-green algae. It matches cyanobacteria like whoa. And what's cool is that cyanobacteria are still around today. The scientist who figured all this out, and we figured this out in a chloroplast before we figured it out in the mitochondria, but the scientist who figured this out is a woman named Lynn Margulis. And she was at UMass, although she did not do this work at UMass. She did this work while she was at Cornell, but she was UMass's one member of the National Academy of Sciences, I believe. Or one of UMass's members of the National Academy of Sciences. She died, oh gosh, she died like about 10 years ago, but she gave a talk at HCC, and I took my class to it, and it was really cool. She was brilliant, and she's the one who actually figured all of this out. So it's actually endosymbiosis that was first formulated through her work with the chloroplast. And we will return to cyanobacterium when we're studying photosynthesis, because they're really cool. Cyanobacterium, they're also known as blue-green algae. I mentioned that already. But they are probably the most sophisticated prokaryotic cell out there. It is so darn close to being eukaryote that you'd think it graduates sometime, but it doesn't. It doesn't have a nuclear membrane, so nope, it's not. But it's so sophisticated. It's like a living chloroplast. Really, really cool. Yeah. So we talked about that. We talked about chloroplasts. Let's finish up chapter 3, and we'll talk about some endoplasmic reticulum. And why don't I draw? I'm gonna go back. Here. Let's get out of green because that's so chloroplasty. All right, so we got our eukaryotic cell. We got a nuclear membrane. You got the cell membrane. Got a nuclear membrane. Got the nucleolus there. You've got all of these mitochondria. You've got some free-floating ribosomes, but a lot of ribosomes are actually membrane-bound to the endoplasmic reticulum, which is just a series of folded membranes in the cell. That is the endomembrane system right there. And it's made up of the smooth endoplasmic reticulum, the SCR, the rough endoplasmic reticulum, and the Golgi. The smooth ER is involved in lipid synthesis. Whereas the rough ER is all about protein production. And it's all about protein production because the rough endoplasmic reticulum, and this is why it's called rough, is because it is just dotted with ribosomes, ADS ribosomes. You don't have an endomembrane system in prokaryotic cells. The Golgi, on the other hand, modifies proteins. And this endomembrane system is only in eukaryotes. Right there. So this is the squiggly stuff over here. That's the smooth endoplasmic reticulum. This is the rough endoplasmic reticulum. And oh, I did not do a good job of drawing the Golgi. But it actually packages up proteins in a vesicle, modifies proteins that it receives from the ER, rough ER, and modifies them, packs them up into a vesicle, and basically sends them outside the cell, like we saw with exocytosis. Okay, I think that is it. That is it. We are done with chapter 3 now, and we will move on to chapter 4 next time. Thank you for bearing with me, for chatting with my dog and listening to my neighbor's lawnmower for just a little bit. I hope you're well, and we'll see you next time. Bye-bye.